Good afternoon, everyone. I guess I need to introduce myself first. My name is Enfu Hui from UCSD. It is my great honor to introduce Erica today, who I guess many of you know is the CEO and president here. Okay, so some background. Erica obtained her BA from the Rice University with double majors, biochemistry, cell biology, and evolutionary biology. She did her PhD with Ian Wilson and Scripps, working on several topics, structural biology related, especially the crystal structure of HIV neutralizing antibody. And she did postdoc training in immunology with Dennis Burton at Scripps. And in 2003, Erica became assistant professor at the Department of Immunology at Scripps, and she was promoted to associate professor in 2008 and full professor in 2012. And in 2019, Erica left Scripps and joined the Jolla Institute for Immunology here. And in 2021, she became the president and CEO of the Institute. I believe she's the fifth president of the Institute and also the first female president of the Institute. Erica is extremely productive, has more than 170 publications to date. She was the first to discover the structure of the Ebola virus surface glycoprotein, and she also predicted that the Ebola virus receptor was located in the endosome rather than on the cell surface. And later, she showed that the Ebola virus VP40 matrix protein can fold into multiple distinct structures. Her lab also discovered structures of many glycoproteins of several other viruses. And Erica is not just an amazing scientist, but also a true leader. She has served on many leadership positions. Just mention a couple. She's a director of the Coronavirus Therapeutic Consortium and director of the Viral Immunotherapeutic Consortium. Erica received many honors. She's a fellow of American Academy of Microbiology. She also a fellow of AAAS. She received the Gallo Awards for scientific leadership in medical virology in 2018. She was the San Diego Scientist of the Year in 2021 by the ARCS Foundation, and previous research hero in 2024, this year, by the Conrad Foundation. Finally, Erica is not just a brilliant scientist, but also a high-achieving athlete. She was an avid rugby player throughout college and graduate school. And she also toured twice with the U.S. Women's National Rugby Union team as the manager. She was not just a player, but also manager. But we are all blessed that Erica chose science over sports. So today, Erica is going to share with us how to use neutralizing antibodies to fight virus. Thank you, Erica.
Thank you. How's the volume? Is it good? Good, okay. So today I'm going to tell you new stories on an old virus. Everything I'm going to show you is unpublished. So this is a story about measles virus. And measles virus is such an old threat to the human population that it has a different name in nearly every language. It probably spilled over to human populations around 500 BC as a divergence from rinderpest when we began living in cities and living near cattle. So measles virus causes more childhood deaths than any other vaccine-preventable disease. And it's also one of the most infectious viruses we know. So virologists use this term R-naught, and that's how many unvaccinated people can be infected from one infected individual. And so for measles, it's extraordinary. It's 12 to 18 people. And it was a virus that we thought we had eliminated in the United States. And elimination of a virus means we've gone 12 months with no new cases. However, it's come back. It's come back every year, more and more every year. And in fact, this year already in 2024, which we're not quite halfway through, 17 states have had measles virus outbreaks. So why is that? That is because it is such an infectious virus that you need at least 95% vaccine coverage in order to achieve herd immunity. And we have now fallen below that number. So we don't really have effective herd immunity in this country anymore. And there's a couple reasons for that. Some are sociological. People don't want to be vaccinated or vaccinate their kids. And there's also biological reasons. And the biological reasons are that the vaccine we use for measles virus is a live virus, is a live attenuated virus. And so it cannot be given to the immunocompromised. It cannot be given to pregnant women. And it is not given to children before the age of 1 because since it's a live attenuated virus, maternal antibody that the babies likely have may inactivate it. So in a case where the baby is going to go off to Europe or some place where they're likely to encounter virus, you can vaccinate them younger, but typically all of our infants are vulnerable. And the other interesting thing is that about 5% of people who receive the full dose don't seem to respond, and they don't seem to develop levels of antibody. We don't really know why. The issue now is that vaccine experts like UNICEF and WHO are predicting kind of a perfect storm of conditions where we've fallen below the threshold of herd immunity. We have more and more people living with diseases of where they're immunocompromised that would not have been alive before, coupled with societal events that are increasing the number of vulnerable people. So because of the pandemic, 20 million kids missed their regular vaccination program. You can predict that some of them fully have caught up. Also, anywhere there's displacement, conflict, refugees in the world, disease spreads. And as even those of us that did get vaccinated get more and more years from those doses, our immunity may be waning. So people are predicting that we are ripe for growing numbers of measles virus cases every year. So is that a credible threat? It very well could be. We don't know. There are more and more measles deaths. In fact, there was a child that died of measles a week ago in Canada. So it could be. But what treatments do we have for someone that presents with a measles virus infection? We've known about measles for a couple hundred years. We have sophisticated virology tools. We have nothing. There is no specific treatment, even now, for measles virus. And so that is where we thought there were some things that we could achieve with the tools that we have. So what my laboratory wanted to achieve here were 3 different things. A basic understanding of how the virus gets in, what immune recognition looks like, what's successful, what's not successful. Can we use that knowledge to make antibodies against the surface glycoproteins that you could give someone if they had become infected as a treatment? And also to try to develop a novel subunit-based vaccine for people that cannot receive a live attenuated vaccine. So those are our objectives. Now the targets would be the surface glycoproteins on the virus. Now a lot of viruses that we know and love, like SARS-CoV-2 or know and hate, Ebola, HIV, they've got one glycoprotein on the surface. And that one protein does both the jobs of attachment to the target cell and fusion inside that target cell. Measles has 2. It has a hemagglutinin that does the job of attaching to receptor, and it has a protein called F for fusion, and that's what does the job of fusing the virus into the cell so that the viral membrane will merge into the human cell membrane, create a pore, and that allows the viral genetic material to enter the cell. And these 2 proteins have to work in concert. Receptor binding to the hemagglutinin causes some transmitted event to the fusion protein we don't yet understand, which triggers the fusion protein to change conformation such that it will drive fusion of viral and host membranes. Okay. The process is probably something like this. On the surface of the virus, that fusion protein is a trimer, a 3-part molecule, and it exists in an unstable structure we call the pre-fusion, before fusion structure. In response to some trigger, it first goes into what we call the pre-hairpin intermediate. And that's when hydrophobic fusion sequences will unwrap, drive themselves into the target cell membrane, and anchor in there. That's the first stage. Then there's a refolding event where this polypeptide and that polypeptide start to collapse to form a very stable 6-helix bundle, which changes the shape of the rest of it. This is a very stable molecule. That is an unstable molecule. So this whole pathway is an energetic roll downhill. And if you make the wild-type protein by itself, all you get is this in the post-fusion structure. So the events that drive measles into the cell are something like that. But what do we know about that process at high resolution? What has science actually observed to this point in time? Just that. That's the only structure that we have thus far. And that structure was the Ph.D. thesis of Takao Hashiguchi in 2018. Now, Takao went from there to our lab to do a postdoc where he was the one that solved the structure of the surface glycoprotein of Marburg virus. But that was his Ph.D. thesis before he came. And it was really hard to get that thing to crystallize. And he had to do a lot of gymnastics, I'll show you. But the way that measles protein is organized is kind of like this. So it's made as a polypeptide precursor that is cleaved by furin. It's a little bit counterintuitive that subunit number 2 comes before subunit number 1. Doesn't matter. The way that's organized in the structure is like this. It makes this strand from southeast to northwest. It coils up into a globular domain. Another strand that reverses direction coils up into a globular domain, and then there's the stalk. And 3 of those twist around each other like a soft serve cone. And the fusion peptide, the little hydrophobic sequence that's tucked up in here and unwraps and drives itself into the target cell membrane to make that prehairpin intermediate, is tucked up in here. Okay. And in order to crystallize that molecule, what Takao had to do was add in 4 disulfide staples. He had to engineer in new cysteine residues that would anchor the stalk together. And he solved the structure by itself and with a lot of different small molecule inhibitors of fusion. These aren't really good enough to be drugs. They're good enough to get a structure. So that's what Takao had to do. And we were kind of interested in this whole cascade of events. And what are the structures that change here? And how does the immune system see the evolving antigen? We wanted to understand that transition. And to understand that transition, we needed a version of that fusion protein that could change conformation. It wasn't stapled in place with the cysteines, but not too much and not too fast. We wanted it to go to the end, but not before we could get its pictures. So what we had to do was a couple of things. Now, this is work of postdoc David Jolla in my lab. So he couldn't use any engineered disulfide bonds. That's a trick a lot of structural biologists had used. Instead, he was pretty clever. And working with our collaborator, Matteo Perotto at Columbia, they looked at a series of point mutations that were associated with measles virus that caused fatal infections of the central nervous system. And in that series of mutations that emerged, there were some that stabilized the protein and some that destabilized the protein, and they'd always kind of coexist. Well, he picked 2 of the ones that stabilized the protein, and that was enough to hold the molecule in this pre-fusion shape for a while. About 85% of the particles he first expressed were pre-fusion. If you wait long enough, it'll go to post-fusion. And so that was what he was able to do, is get the cryo-EM structure, the first cryo-EM structure of the post-fusion protein, as well as a natural sequence of the pre-fusion protein. And there's a couple of scientific firsts I want to show you here. The first one is this resolution. This is the highest resolution structure of any viral glycoprotein ever solved by cryo-EM. So go team LJI. The second thing I want to show you will come in a minute. So I want to show you how it changes conformation. So this is how it's organized. So there's a strand southeast to northwest. There's the globular domain, reverses direction. There's the globular domain, and then there's the stalk and the tail, just like Takao saw, and the 3 of those twist around each other. Now that we have the pre- and post-fusion structure, and this is the other first, this is the first sequence-matched pair of these structures for this whole virus family. So you can now morph the beginning to the end to see what the conformational changes are. They are these. So this is the morph of the larger F1 subunit, and you can see it flipping up to adopt the pre-hairpin intermediate, and this sequence is going to become the long polypeptide stalk with the other one. If you add in the F2 subunit, it kind of stays still and goes along for the ride, and you can see the conformational transitions that happen between pre and post-fusion. All right, so we understand the beginning, we understand the end, we can model the transitions in between. This is probably a relevant antigen for the immune system to target. If we know antibodies against those, you can deliver them as post-exposure therapeutics or pre-exposure prophylactics. Are there antibodies known that will neutralize this molecule? Precious few. But here's one, in this paper here from Matteo Perotto. It's called Antibody 77. Matteo, working with Stefan Nivist at Ohio State, evaluated this antibody and compared it to a peptide, synthetic peptide that kind of blocks that 6-helix bundle formation and one of those small molecules that's kind of moderate in activity. And they show that it can neutralize in cell culture and it can protect in a cotton rat in vivo model at low dose or a high dose. Okay, so it's got some effect in vitro, it's got some effect in vivo. That was enough for it to be interesting to us. How does this antibody work, and what does it do to interrupt that process? Enter cryo-EM. So this is our work, this is what we do. This is a photograph of the individual protein complexes. So if you see from the back of the room these little triskeletons, these little trimers, these are each individual copies of the fusion protein with 3 antibodies stuck on them. The way cryo-EM works is we take our proteins in solution, we pipette them onto the grid, we plunge freeze it, and then we have photographs of individual protein molecules in every possible orientation that have all fallen on the grid. And we can take all the ones oriented like this and average them together, all the ones oriented like this, average them together, all the ones oriented like this, average them together, and then computationally create the 3-dimensional structure that became that. So we begin with 2-dimensional classes, and you can start to see some structure emerging here. So this is all the particles that were oriented like that, individual protein molecules. And eventually we can reconstruct 3-dimensional structure. So this is now the cryo-EM structure of the measles virus fusion protein. It's in its beginning pre-fusion conformation, bound to this antibody called 77. So here's the whole FAB fragment. We're modeling in the business end, the variable half, heavy chain, light chain, and you can see where it binds. And it's actually done something kind of interesting. So here's the surface of the fusion protein. Here are where complementarity determining regions, the CDR loops, of interest are binding, 3 from the heavy chain, 1 of interest in the light chain. And what it is that they interact with, all the different hydrogen bonds and contacts that they make. And we can see what it does is it interacts with 3 different pieces of polypeptide that all come together in that structure from different subunits. So it sees a piece of the F2 subunit from here, a piece of the F1 subunit from here, F1 from a different monomer, that's the fusion peptide, and here's another bit of the F1 double prime subunit. So 3 different pieces of polypeptide that come together from different monomers in the trimer, as well as a piece of the fusion peptide. So the fact that this antibody recognizes things that have only come together in the pre-fusion structure explains why it is that we see this antibody only interacting with the before and not the after. This antibody can only bind F when it's in its pre-fusion conformation, can't bind it once it's gone and sprung into the post-fusion. Okay. So that gave us an idea about what its mechanism of neutralization might be. If you have an antibody that recognizes things that have come together and it's anchored them on, how might you expect it would neutralize? Keep them from coming apart, right? This is what we're expecting from every other antibody that we know against that kind of protein. But Matteo actually went and did an interesting experiment where he has an assay that can take apart all the different steps and find out where the antibody arrests. Does it prevent attachment of H? Well, no, it's an anti-F antibody. Does it prevent the triggering to the pre-hairpin intermediate? Or does it prevent the full-on fusion? And what we were sort of expecting was that it might prevent triggering. We thought it would prevent anchor on here to keep this thing from changing shape so that its fusion peptide didn't go and insert into the target cell membrane. But we find actually that what it does is it arrests it here. Somehow that antibody allows the fusion peptide to unravel and the fusion protein to adopt its pre-hairpin intermediate there. we can see that the fusion protein has irreversibly anchored onto the target cell despite the presence of the neutralizing antibody. And that was kind of a real head scratcher for us. How could that be happening when we see in its structure that it's bound to the 3 different pieces? There it is. It's clearly specific for the pre-fusion conformation. And so this became kind of the beauty of cryo-EM for me. We went back to the grids. Remember, in our grids, when we do our cryo-EM experiment, we put all of our protein solution down onto the grid and we plunge froze it. So we see pictures of all the individual protein molecules in whatever shape and status they are. And then we're using the computers to pull out the ones that are oriented like this, the ones that are oriented like this, and averaging them together to compute the structure. And we went in looking for the antibody fusion protein complex. This is what we're trying to pull out. So then Davin went back to his grids and I was like, well, what else is there? What have we missed? And so here are the structures that he saw in the grids. Now this is the structure that I've shown you before. It is the fusion protein bound to 3 different FAB fragments. And he's helped you out by coloring the different domains, different colors. He's got 140,000 protein copies that look like that. They gave him a 2.5 angstrom structure. He also saw a couple thousand molecules that look like this. It's also a trimer. That's F, but if you look at the center, it's different. These orange pieces have come together in this tube in the center. They've unwrapped to try to start making that helical bundle in the center. I had a couple thousand particles that look like this, where one of the monomers has started to fold away, and it's left with 2 bound onto their domains. And then he had 96,000 particles that looked like this. And that's just an FAB hanging on to 2 different domains. Because he had 96,000 copies of that, number one, that tells you something has arrested at that point, but he had enough to get a structure. So he could get a 3.6 angstrom structure of that here. Now here's the original pre-fusion. Here's the antibody. Here's the heavy chain and the light chain. And you can see it binds on to turquoise and green and a bit of red. What it's left hanging on to after the thing has been allowed to unravel is still the turquoise and green and red, but the fusion peptide's gone away and the rest has gone away. So it's actually able to directly image the conformational intermediate of what it's arresting. So the antibody can only bind here. It can only bind F in its pre-fusion state, but it allows it to begin to unravel, to anchor into the target cell. It allows one to fall away and start to separate, and then it's hanging on for dear life here, keeping 3 pieces together still at the center to prevent the fusion from going all the way to the end. So we've started to model what that might look like. So here we've colored the fusion protein by its different domains, and you can see the fusion peptide here is going to penetrate in the target cell membrane. And then we're going to allow the conformational change to occur whereby these unravel, you start making the 6 helix bundle, and the action of making the stable 6 helix bundle with blue and orange together pulls the membranes together and it drives the fusion. That's what we think happens. Now watch again up here. In the turquoise, the green, this is where the antibody binds. If you watch that site, this guy falls away. Watch them rotate. See how they rotate relative to each other as that begins to uncoil. If we add on the antibody, now unfortunately the antibody is colored the same color as the turquoise here, and this is the FV, the business end of the fab, not the whole thing, but you can see the bulk of the coils there. It allows the fusion peptide to fall away, but it's going to limit the rotation and the freedom at the top. And probably the steric bulk of the thing, because remember it's not an FV, it's a whole IgG in a biological scenario, is going to block those 2 membranes from fusing. So we're actually able to directly image an antibody that has arrested something in the middle of this transition. That's really cool. And what's really cool is science had never really seen this kind of thing before. Structural biology has always shown us this. And that is because, until very recently, if you're going to see a structure, it had to come from a crystal. And so our textbooks of what proteins look like are the images that came from crystals. And if it came from a crystal, that means it was something that could form a crystal. It was symmetric. It was fully occupied. And so the understanding of structural biology and what glycoprotein antibody complexes look like that we all learned on our mother's knee comes from things that would adopt complete occupancy and beautiful symmetry where all the particles were the same shape. Biology is not really like that. Biology is a mess. These proteins are heterogeneously glycosylated, where some positions have the sugar and some don't. These things adopt multiple different conformations. The antibodies are sometimes in full occupancy, and sometimes they're 1 or 2 or 3, and sometimes they're asymmetric. And sometimes that's really important for understanding what the right surface is for antibody recognition. And so this is what we all grew up seeing, and this is our expectation of how things happen. But now we have the tools to start to look for this. And what you needed was someone like David that would dive back into the mess on the grid and look and see what else was there. Because if you looked at that little particle left of one fab attached to a piece of junk, nearly every other structural biologist would throw that away. I think that's junk. That's not what we're looking for. We're looking for the trimers. But he found it and gave us a new insight. And so because of all the different firsts, this work is now in press at Science. So I've introduced you so far to the first structure ever of any antibody against measles. Please meet the second structure of any antibody against measles, this one. So David, working with Jolla Institute, a new graduate student, just solved this one. So this antibody, also from Matteo's lab, is different. It doesn't bind at the bottom. Remember, this guy rests somewhere in the middle of triggering. This guy binds at the top. One FAB fragment, light chain, heavy chain, nails in the top of the apex of that trimer, simultaneously interacting with all 3 monomers. That's another kind of thing that was never going to crystallize, and it evokes the new structures that we've seen of a really potent antibody against Lassa does that. A really potent antibody against Ebola does that. We've got another one in this antibody Y10F. Please meet the third structure ever solved of an antibody against measles virus, H8. He also binds at the bottom, but what's different about this structure of F versus these structures of F? Do you see it? Pre-fusion, post-fusion. This antibody actually drives F only into the post-fusion state. We cannot get an image of this in the complex with a pre-fusion particle. So the way this antibody neutralizes to accelerate the triggering of the molecule before it can ever get to the target cell. So in 3 structures, we've got 3 different mechanisms that are all fascinating. Now, that all comes from analysis of antibodies from mice that were elicited by Matteo Perotto's lab at Columbia. Enter LJI and what we're good at. So let me show you now the first panel of human antibodies against measles virus. And this is the work of Marissa Acchiani, new postdoc, working with Kate Hasty and Stephanie Harkins. So from a single person with samples left over from the COVID work a couple years ago, and the single person had been immunized against measles, Marissa has gone with a discovery campaign, and she's now got 60 new monoclonals, human monoclonals against H. And here are 130 new monoclonals against F. So we have twice as many against F because the literature says that 80% of the antibody response will be against H, and that antibodies against F are weakly neutralizing. So we thought we might have a hard time finding antibodies against F. We went in there twice. Wasn't true. I think the literature says that antibodies are predominant against H, and they only neutralize against H, because people had a really hard time making good F before. If you're not a protein engineer, the F you make is all post-fusion and aggregated and pretty crappy. And so antibodies you could pull out with crappy post-fusion F are indeed not neutralizing. So I think that's just a bias in the literature with the tools that are available. So we call this kind of figure the Death Star figure. And what we are showing you here are the diversity of VH gene usage. So we've got lots of different solutions for binding lots of different places on F. And a plot of the affinity. So some of the best ones are 20 picomolar. And what we're starting to do now is to use our Cartera LSA to map the binding communities. Do they bind different epitopes? How much do they overlap? And we can see already that MRSA has 7 major communities or competition groups of antibodies against H, for example. So if you take that one, we're starting to image it. And so in addition to working on measles, we've now opened a French bakery on the 2nd floor. They look like croissants, but they are actually structures of H as a dimer bound to 2 of the FAB fragments. So we're starting to map the structures and recognition patterns of all these human antibodies against measles in lots of different ways. And what you can look forward to in the next year is exploration of all these different antibodies and understanding of what the immune response is against the virus. And that'll guide vaccine design. Now I want to show you one more story. I think I have time for that. And that's something new that we've just done. So the first thing was a new story on an old virus. This solves an old problem of a new virus. And this is structural biology inside the cell, inside an infected cell, of the Ebola virus nucleocapsid. So these viruses that we work on are super simple. So you and I have, 20,000 genes, like 20,000 functions. These viruses have just a handful. So Ebola has only 7 genes. And over the first 20 years of the lab, we did a whole lot of different crystal structures of the individual pieces by themselves, we and others in the field. And we've recently been able now, with the new tools we have, to start to put these things together. So we had a box of Legos. How do we put the Legos together to build a model of what the virus actually is? So Ebola virus, this is this long filamentous virus. And if you look inside that virus, this is now a watercolor from David Goodsell. At the center of that filament is the nucleocapsid. It's like an old telephone cord that spirals up and up like that. That is built by repeating copies of the nucleoprotein called NP, nucleoprotein, build in a spiral along the viral RNA genome. And as the NP builds, it scaffolds things that it needs. So there are 2 proteins that it needs. One is a viral protein that's 35 kilodaltons big. One's a viral protein that's 24 kilodaltons big. That's how they got their names, VP35 and VP24. Genius. Here's what we knew about NP. We knew it was a polypeptide that kind of had 2 halves. The N-terminal domain was what you need, all you needed to build that kind of tube. And the C-terminal domain, no one had ever really seen. It's mostly intrinsically disordered. Really don't know what it looks like. But that N-terminal half is all you need to make that telephone cord and the nucleocapsid in the center. We had solved its crystal structure. In order to make it behave well, we discovered that we could take a little piece of one of its friends, VP35, and use that little orange piece to do what we call a chaperoning, VP35. So a chaperone at the school dance keeps the molecules separate. Chaperone here keeps this NP from polymerizing. And that is what we crystallized, and how it relates to what we think the whole biological assembly is something like this. The VP35 is some kind of a oligomer, and then NP has these disordered tails. And we thought, based on the information that we had, that the job of this complex was to keep that molecule single until it could be delivered to the assembling nucleocapsid. So it kept it single, it kept it RNA free. And we thought that its job was just to get it to where the genome is, so it wouldn't polymerize erroneously on cellular RNA, but instead polymerize on the genome, like that, like so many ballet dancers on their bar. And we do know that as NP polymerizes along that RNA, it clamps down on the genome, so it changes conformation. So it's kind of like, the 2 halves of a Ziploc bag coming together to anneal. So this is kind of a different shape of NP, this green peanut, from all the ballet dancers on their bar. Okay. Now, what do we know about the whole polymerized thing? This is a structure from the University of Tokyo of recombinant protein. So it's just the ordered N-terminal half polymerized on cellular RNA. So it's not specific, it'll bind any RNA. Here it's polymerized in cellular RNA. Here's 1 copy, so that's 1 peanut, and it's just polymerized like that. Beautiful structure. That's recombinant proteins. The work we had from virus comes from John Briggs' lab at the LMB in Cambridge. And that's his image of the whole virus. And if you look at 1 copy, that's 1 peanut in pink, and you can pull it out, you can see here all the different ballet dancers on their bar where the RNA genome is in yellow. Now if you take that and you rotate it on its side. So now here's the genome, the ballet bar. Here are the copies of NP ordered like this into the screen. You can see that the nucleocapsid has all of its friends scaffolded on the outside. Now here started the puzzles. That made a lot of sense, but that's just one protein polymerizing. This started to be confusing. We got that the NP was clamping down on the genome. Fantastic. This was weird. It was weird because these are the copies. We can see in the structure these are copies of VP24 scaffolded on the outside. But it was weird because one was built pointing in, and the other one was built pointing out. And that's just odd, right? Normally, if there's an AB protein complex, it binds one way. It doesn't bind equally well this way. So the field did not know what that was. It was just odd. And there are a couple other mysteries, like what's that? And what's that? Couldn't tell from the density at the time, but it was thought it was probably that floppy C-terminal half of NP that no one had seen before. Maybe it filled in there somehow. OK. And the other mystery in the field, kind of some biochemistry we've been doing, when we've been playing with VP35, has had two independent binding sites for NP, and they were both equally functional. One was the chaperoning site, and one was somewhere else. So why would have two independent binding sites? We didn't really know either. OK. So those were all structures that were either recombinant protein or the final virus, just John Briggs imaged the virus. We were kind of interested in the process of how the thing assembles inside the infected cells. And doing structural biology inside the cell is what I feel is the frontier of imaging here. Until recently, structural biology was only ever done on recombinant protein. And that was great. We got structures. But proteins don't exist by themselves, especially not in virology, right? The viruses can't live on their own. They have to hijack all kinds of things from the cell. They're inactive unless they can use all these cellular factors. They need their own partners from the viral genome in order to accomplish their functions. And we know these proteins are multifunctional. They're different structures in different places at different times, depending on what cofactors are there or not there. And if you're doing a recombinant protein experiment, and it needed a certain factor in order to adopt a certain shape to achieve a certain function, well, if you didn't know that factor existed, you didn't supply it in your recombinant protein experiment. And so looking at recombinant protein alone was kind of unsatisfying for understanding how these things operate in the cell, what the cell biology of infection was. And so what we set out to do was to start learning how to solve structures inside cells. So this is the work of Reika Watanabe in the lab. So this is what we do. We grow our cells on these little tiny copper waffles. They're like, 3 millimeters across. Actually culture the cells on the dish. We plunge freeze them, so we have frozen cells. We use a model of Ebola virus with a GFP so we can find the infected cells. We use a gallium ion beam to make a thin section so you can mill off the top of the cell, mill off the bottom of the cell, and you get a thin section of the cell to image. You can put it in a microscope and you can start solving structures of what was inside the cell. So this is a tomogram, so it's all the different images of that thin section of the cell taken at different tilts, built back together into a three-dimensional volume. You can see the arrow going up and down in what looks like rattlesnake rattles. Those are actually the building nucleocapsids inside the infected cell. And we can see, because it's in the process of assembly, we see some that are loose, some that are condensing in places, and some that are fully assembled on their way out. We can actually see the processes of assembly. So then we can trace what the nucleocapsids are in the cell and start to average them together to build the structure that was there. And after enough particles and enough tomograms, we get something like this. So this is now the structure of the nucleocapsid being built by infected cells at 9 angstrom resolution, kind of imaged from the cells. Now, as it rotates around, all the density is colored the same color. It's all this peptobismuth pink. On the inside, super easy to see the rungs, right? The ladder rungs, those are the rings as it builds up in the spiral. And the outside, you can see all the scaffolded stuff. So there's the inside, here comes the outside. Lots of things carried around on the surface and the outside. So Reika can start to read from the shape what goes where. And she can actually build what all the different density belongs to. So this is one building block, one repeating unit, one NP peanut with all of its friends. And she can show you what everything is. So here, in blue and turquoise, are 2 copies of NP, that's the basic repeating building unit, on this magenta RNA. Those are the ballet dancers on their bar. And she can identify all the pieces of density. Okay, now this is where it got cool. This is where the field was stuck in 2017. We have the copies of NP, and they're like this on each other. We have the VP24 pointing in, the VP24 pointing out. We don't know why it does that, but it does. We have the unknown stuff. The shape of the unknown stuff was suddenly very clear to us because we had been crystallizing these proteins for 20 years. And we could see very clearly the shape of this was our chaperoned NP in its own conformation. And it fits perfectly. And this VP24 NP interaction surface is identical to this VP24 interaction surface. And that starts to explain why one points out and one points in. This was a piece of the VP35 C-terminal domain. And so this is the end, and that's the beginning, and the protein in the middle is probably disordered. But we don't know if these two connect or this guy connects to its neighbor. But suddenly, we have filled the density that was just a puzzle before. And so we can build the whole molecule. So that's how we differ in virus and cell. Also, because we're looking at the nucleocaps at different stages of assembly, we can see how the thing condenses. Now let me go over here to make sure this is going to work for me. So you see how those molecules are all organizing in concert. I'm going to go back. Okay. So this is the side view of the nucleocapsid. Here are the rungs. Here's the stuff scaffolded on the surface. You can see it here, repeating rung, rung, all the scaffolding network. And then here is the front view down the center, and you just see the scaffolding network because the NPs are on the inside. And if you compare in cell to in virus, you can see how the domains tighten and reorganize. And now if you're looking at the front view, watch how they rotate. Watch orange related to orange. They're perfectly in sync. So as the molecules tighten and condense to build the full nucleocapsid, we can see how maybe tethering the top orange, which is the N terminus, to the bottom orange, the C terminus, might be kind of a wrench that holds them all together. And we were tossing around a hypothesis in lab meeting this week that condensation could change the density of the particle, which affects its fluid mechanics from the replication site. So what we have now here is from LJI, the first ever complete model of the Ebola virus nucleocapsid, where we can see repeating copies of the core of the nucleoprotein. We understand why VB24 points both in and out. VB24 is immunosuppressive. It keeps the infected cell from knowing it's been infected so the viral replication can get going. So covering the NP with repeating layers of VB24 could deliver an immunosuppressive payload. We see for the first time that there's a third outer layer of the nucleoprotein, which isn't bound to genome, but serves another function. In fact, we think it's the tether to the membrane. And we see how it's chaperoned and built together. So it's incredible to me that we now have the tools for science to actually solve biologically relevant structures inside cells. This is always what I wanted to do. What's exciting to me about the section of work I just showed you from my lab is this. All of the things I've showed you, all these different techniques, looking at heterogeneity, discovering antibodies, mapping their domains, doing relevant structural biology inside cells, going from antigen engineering to antibody discovery, going back to antigen engineering. Every technique I've shown you today, my lab was not doing until we moved to LJI 5 years ago. That's only a fraction. We didn't do flow cytometry until we moved to LJI. We didn't do the beautiful cellular microscopy until we moved to LJI and could work with Z. So this incredible array of tools and facilities and opportunity here are really, really special. We did fine before. We were a pretty successful lab. But the people that I brought here have blossomed since moving into the building. And that's from a couple of things. That's from the incredible facilities and cores and opportunities we hear, as well as the culture that really celebrates discovery and working together. So it's been a really special place, and that is why I work so hard for this institute. It's because I understand how very, very special it is. So I'd like to thank Reika Watanabe for all of that in situ cryo-ET. The measles cryo-EM was driven by David Jolla, being helped by a new graduate student, Gail Niemeyer, and our cryo-EM director, Ruben Diaz-Avalos. Measles antibody discovery led by Marissa Akiani, working with Stephanie Harkins and Kate Hasty. And I'd like to thank everybody in my lab and our collaborators at Columbia, UCSD, and Ohio State, and especially the LJI facility directors. The fact that facilities and operations here fixes everything beautifully, everything is beautifully maintained, everything just works. You can just expect to do your science. If something needs to be built, it's built better than specs. The fact that we have a clinical core where you can just get all the human samples you need is remarkable. The fact that everything is so seamlessly integrated, and that the mission and the spirit of the place, that you go to Z and you say, I think I'd like to do this, and he says, we could do that, but we could also do something much better, which is, oh, that's amazing. It's been a really, really special place to work. And I think it's pretty cool that just under 5 years of moving in, I can give a talk where everything I've showed you was something we were not able to do until we moved here. One of it's in press and science, the other's being revised for cell. So it's just been a remarkable place for us to be here. Thank you for welcoming my lab, and I'm happy to answer any questions.
Structurally, how would you get at this non-stable triggering of F by H in measles virus or any other analogous fusion protein triggering that would be a directly in the complex?
You gotta go a bit beyond the expectation of taking still snapshots of things and try to get the idea of movies. So you can do time-resolved EM. And for things that behave well, time-resolved crystallography, where you can take snapshots of things in the act of transition. So you could introduce the proteins and take multiple pictures in the act and try to capture enough molecules. You can also do things like the tomography of the frozen cell surfaces and the immune synapses that we're doing, where you would hope that over enough cellular interactions and enough copies of the particles, you can capture the different things in the different states and try to build and model that movie. You can complement that with spectroscopy, or things that would measure, the distance between this signal and that signal. So you can measure the timing and the space of whenever the different amino acids move together. It's a new frontier. It's one that we want to go into.
And could you distill it even further? Like, for instance, could you put membrane proteins in a liposome and have a recombinant receptor as well, or something?
Yeah.
To try to get away from the whole cell.
Yeah.
Okay.
That's what we want to do. Yes, sir.
Yeah, first of all, I want to say phenomenal talk and congratulations, and also show the strength of LJI. Teams could work together. So 2 questions. One is about the pre-fusion protein. So since it's not stable to start with, so I assume it will test many combinations.
Mm-hmm.
So which one would lead to the functional one?
The beginning structure isn't static. It kind of breathes a little bit, and different viral surface proteins, different viruses breathe more and more. They are all functional. They can all do the job of receptor binding. Sometimes receptor binding introduces an intermediate conformation. Sometimes it's the trigger for the whole thing. So that is functional. Once it flips all the way to post-fusion, it's kind of become non-functional for a lot of different functions. All it's done is fuse the different membranes together. Does that answer your question, or right?
Yeah, so what you're saying is that even though it's testing different conformation, all would lead to it. Right?
Mm-hmm.
Okay.
Yeah.
And the second question about the intracellular structure, that's very cool. Does it depend on the concentration of the protein? Meaning, can we do only viral complexes, or can we look at cellular complexes?
Size helps. So it'll be really hard to find something that's 20 kilodaltons in a cell. Something that's big, like a nucleocapsid, helps a lot. Things of which there are many copies will help you find the examples that you want. So it depends a lot upon size and frequency and order and abundance. If it's something repeated, you'll have more copies to try to average and a better chance of trying to find it. Yeah. I want to point out in the back of the room my husband and our younger son, Beck, who came to hear the talk. And Beck is available for lab jobs and research projects in 2 years. Yes, sir.
I was curious about, you mentioned for the first antibody, the Measles antibody, that you thought perhaps steric hindrance was
Uh-huh.
IgG. So in the arrest assay,
Uh-huh.
is that done with a Fab or a full IgG? Do you think it would change the step at which the arrest happened?
Yeah, but he did it with IgG. Did Matteo also run those assays with Fab?
No, they haven't done it with FOB, but the structure is from the FOB.
Yeah.
So we know it's the same.
Yeah.
Beautiful work. Congratulations.
Thank you.
I have a question about the chaperones. Is the cellular chaperone in the host cell that hijacked by the virus? Does the virus use the chaperone for packing? What's the function?
You mean the orange protein?
Right.
It's not a cellular chaperone, it's a viral protein. And so the term, the use of the term chaperone there is a little bit artificial. It came from chaperone as in escort, like the chaperone at the school dance keeps the couples monomeric. So that copy keeps NP monomeric and keep it from assembling. So it's not a cellular chaperone.
Okay.
Now, viruses do interact with cellular chaperones. A lot of these viral infections induce a stress response or upregulate chaperones. They sometimes use the chaperones to deal with the overexpression of the viral proteins, or they, different things. But this guy is a kind of a misuse of the term chaperone.
And to what extent the in situ in-cell structure differ from the reported Ebola structure?
We see more in cell. That could be that we have better direct electron detectors than previously existed. It could be that it's in cell where things are bundled together.
Yeah.
It could be Reka's talent. The final assembled nucleocapsid structure of the final condensed thing is pretty similar. What differs is that we can see all of the intermediates. We can see the loose coil, we can see the intermediate condensation, we can see the events that have to happen in order to get it out. There is one difference between in cell and in virus. In virus, the nucleocapsid is in the center and it's tethered on this side and on that side to the viral membrane. And so we think that is the flexible, not yet visible C-terminal half of the nucleoprotein interacting with matrix and matrix. Inside the cell, that doesn't need to happen because there's no membrane there yet. So some of those conformational changes are different. Yeah. So sometimes, so many of these viral glycoproteins have a single stalk. So the trimer will have 3 single pass stalks going into the membrane. Often we cut them off and we express an ectodomain. And the expectation is that the part that we see does fit the tomographic envelope of what we see in the virus. So we think it's more or less right. Now what's happening at the very bottom might be different, but often that's disordered in the structure, so we don't see it anyway. When we have reconstituted our transmembrane protein in detergent or micelles or amphifols or something like that, often those transmembrane domains are not well ordered anyway. We just see the ball where the membrane ought to be. So there could be some differences there that we've not fully realized. In the full virus, there may also be connections from the matrix that may help stabilize some of these things. I think thus far it's a pretty good approximation for the parts that we can see, and just an understanding that things may be different in the disordered parts inside the membrane.
Erica, that was fantastic. I guess a bit of a technical question, or just sort of maybe where the field is, structural biology, that animation you showed of the transition to the fusion. So what's the data that drives the
The more.
computationally
Yeah.
determined as sort of a minimal free energy, or is it essentially a merge of a couple of snapshots? Where are you in the field with how to put something like that together?
Yeah, great question. So it depends on the tool. The simplest ones just mathematically calculate this XYZ coordinate to this XYZ coordinate. How do you get from here to there slowly? And sometimes those tools have pieces of protein passing through other pieces of protein to get there, and that's not right. The more complex ones take into account all the biochemistry of how things might hope to move, and take into account kind of some maximum likelihood minimization of how the protein might happen to be. Some of the prediction tools could also do molecular dynamics along the way of understanding, fed in from whatever spectroscopic measurements you might have, understanding actual differences. A lot of what we do is a best guess because no one has ever observed the intermediates yet, and so we're hoping we're getting it right. One of the other challenges is that the computational time required to model some of these transitions, if they happen on the order of many seconds to many minutes, we don't have that much computational time and space available. We tried to model the conformational transitions of VP40 from a dimer to an octamer so it changes its shape to an RNA binding function, and just the time scale it needed to happen could not be accomplished with the computers we could assemble. So we went with XYZ to XYZ, and how do we get there?
Do you see the in situ structure, how scientists identify novel virus interacting proteins?
We could. So that's the big hope, that if the
Yeah.
tools get good enough, you can start to identify, like if there's a blob attached to your protein, what is it? Yeah, and then you can start to do bottom-up proteomics. If you just throw all your stuff on the grid, and then if you can see it, you can start to image it. Yeah. Any other questions? Yes, ma'am.
So much mutation on different strain of the virus can change the different conformation?
It could, very well could, right? It could change how it breathes. It can change presence and absence of sugars, and presence and absence of sugars will change its mobility.
And are you there yet to model that, or do you actually do the experiments to figure out how it's changing?
It could be modeled. We are not a computational modeling lab. Romy Amaro at UCSD has done a lot of that work of looking at what's the glycan shield and how do things move. Yeah, but it could be done for sure. All right, thank you so much for coming. I think there's some other time.
I believe there's a happy hour after this. Thank you so much, Erica.
Hey, thank you for staying for the whole thing.
I was able to stay till the end.
Thank you. Thanks so much for coming. Really appreciate it.